got me the sound TV on it.
I'll be happy. I'd be pretty. We don't have to-
Why does it keep saying it speaking doesn't work?
I feel like it'd
What's
be
wrong?
probably be more better when I was doing the setting for it, so that's probably.
I saw you all day yesterday.
I tried it all day. I tried so hard, but I was sitting there like, that's so much stuff to do this week that I'm like, actually, it's not going in my brain. I know it's not going.
I was like, I have a lot to do like next week, the presentations and stuff, and I was like, and then this exam, and then
You have
bar.
a mission due next week?
Yeah, our presentation is due on Wednesday next week.
Dude, if he moves, he sent out a text saying that
I
he's
seen
gonna
that.
potentially move people. If he moves my group, we haven't even started.
presentation-
our meeting till
He
after
said he's gonna
spring.
assign it for Monday.
Yeah, I swear, he'll do it like Sunday night.
That would be so cute. That would be so cute. because
I'll show.
I was banking on having like this weekend, a few more days to work on it.
Yeah, our group hasn't started because we don't go to
Oh,
after
so
spring
sad.
break. So if we can go, he'll probably tell us like Monday morning.
Yeah. We may need to form next week.
Dude, what's going?
I didn't forget. Do you have like a midterm for that class?
I read ETI under like the test section, but he said we haven't looked at the semester.
It's
But-
so it's not the quizzes that are at the end of the exam.
No, those aren't like worth anything.
But you taking one?
Not the process tests.
I don't understand how is it.
Like someone, you know, like they're under the camera or whatever, and someone like from ETI.
I was wondering like what, we took a T's test on there and it was like a video view.
Yeah,
Yeah.
that's
But
not.
I just didn't know like, I just didn't know like what that means.
I think it's just like what you said. So that's really watching. It's just weird,
But
like.
then it will like notify us.
No, it's fine.
For sure.
Yeah.
They think you're like looking around or
But
something.
then she like.
So intense.
That's really Did you ever find out for the project, like the quizzes on ATI? Are those, do you take them multiple times or are they?
I don't think so.
Okay, so it's just.
From what I've been able to find out, it doesn't look like you can. Because when you put them together. There normally is a box that you click that says multiple times, how many times you want to take it. There's no box like that. And then when I looked at the instructor's instructions and what have you, there's nothing about taking the quiz multiple times. Now, maybe you can, they just don't say so, which would be awfully odd. Wouldn't you agree? My guess is you only take it once. That's my guess. They're 20, but you have plenty of time to take them. You have an hour to take 20 question quiz, so you have plenty of time. So should be doable, I would think. Okay. Okay, and Anna, Stephanie, you guys hear me all right?
Yes, I can.
Good.
Yes, I can too.
Excellent. Okay, great. All right. Okay, let's give this a shot. Oh, wait. No. We're going to, no. Going to watch this one first. Okay, get this stupid commercial done. Okay. All right, so we're gonna watch a video that's all about fluorescence in situ hybridization. And we're also going to look at another video on something called polymerase chain reaction, which is very important for genetic testing. So let's go ahead and watch this.
Hello and welcome to today's talk about FISH, or fluorescent in situ hybridization. FISH is a cytogenetic technique for visualizing specific chromosomal locations. This is done by the binding of a so-called fluorescent probe that is complementary to the specific region of the country. We talk about that in detail in a moment. The aim of fluorescent in situ hybridization is to detect chromosomal abnormalities that could cause disease. Fish is a very useful tool for medicine because it can tell us whether the chromosomal location is mutated or not in an infant cell. The first step of the fish assay is the fixation of the cell with a formalin-based fixation. That causes extensive protein and also protein nucleic acid coupling. After that, we can add our probes. But how are these probes designed? First of all, the probe has to be complementary to the chromosomal region of interest. This can be either done with a short double-stranded RNA probe or a short double-stranded DNA. With an enzyme called DNA, we are able to create random cuts in the DNA. These cuts are also called niches. Now we provide special nucleotides that have a fluorophore attached to them. Ligase can now seal these groups with our fluorescent nucleotides. We successfully created a probe that is labeled with our marker. With PCR, the probe can be amplified. The next step involves the denaturation of the probe that was introduced into cell culture. The chromosomal target DNA has also been denatured. Denaturation can be achieved by heating up the cell at approximately 95 degrees. After that, the temperature is cooled down, allowing the latent probe to specifically bind to the target DNA sequence. Once hybridization is place, all probes that are not bound to any DNA are washed away. The results can be analyzed under the fluorescent microscope. If the probe is designed so that it can only bind that it is 100% complementary to the gene, we expect to see a space location on a chromosome under the microscope. But with any sort of mutation occurred in this gene, no hybridization is possible. For such probes, we can reach an entire enough to fail binding to the site of interest. No fluorescent signals were obscured in this case. In research and specifically in medicine, fish is commonly used to create a karyotype for looking at chromosomal aneuploidy. Therefore, a long probe is designed to cover whole chromosomes and stain them in different colors. Trisomy 21, also known as Down syndrome, can be detected like this. Also, diseases that are caused by genetic mutations can be visualized. Usually, short probes are used for this, since only a certain gene should be observed. I hope that was helpful, and please ask some questions in the comments if you want to know something else.
Yeah, right. Okay, so the URL for that one is on the Blackboard site if you want to revisit it at some point in time. So that is fluorescence in situ hybridization, otherwise known as FISH. I don't think that's going to work because if you don't do this, then very often it will not respond and they won't be able to see the slides moving. Okay. There. Now let's try that. So this illustrates essentially what we tried to talk about last time. Taking a piece of DNA, it can be RNA too. RNA, in fact, is probably a little easier to work with for these types of experiments. When you make them, you use labeled nucleotides that have a fluorescent dye attached to them. You then denature if it's a DNA molecule, you just denature it. That's easy to do. You heat it up. and it denatures. The chromosomal DNA can be denatured using a variety of techniques. In the YouTube video, he mentioned heating it. You can also treat it with alkali. That will denature it as well. And then you use a solution, a hybridization solution, and the probe will hybridize. Any unhybridized probe you wash away. View it in a fluorescence microscope, and you get a little band that has this glowing line on it. When you see this in a chromosome, it's very hard to miss, as you might guess. You can see the outline of the chromosome usually pretty well. The chromosome autofluoresces just a little bit, and then the probe usually will fluoresce a certain color depending upon what dye you use. You get nucleotides that will make the probe glow green, others that make it glow red, others that make it glow yellow or blue or violet, or there's just all kinds of cool stuff available. And then you can label chromosomes with multiple probes and get them to where they're blue, green, purple, you know, red, white, and blue. You can be really patriotic about it if you want. And you can even do multiple chromosomes. And that's very useful for a variety of studies, but for medicine, usually, if you're detecting a gene or something like that, you use short probes and use usually just one color, unless you're doing a multiple gene sort of analysis. And we talked about this already. So DNA analysis can be used to detect certain genetic diseases. Certain states mandate certain genetic tests for every newborn. Michigan mandates 2, I think. Did that change? I think it's just 2, if I'm not mistaken. But I think sickle cell disease is one, and not sure what the other one is. I think it's just 2 in Michigan. So your probe, again, it's a piece of DNA that has a sequence that's complementary to the gene of interest. If you want to just detect a presence or absence, or presence or absence of a gene, presence or absence of a specific mutation, or something like that, you can use a technique called polymerase chain reaction, or PCR. The beauty of PCR, it requires very, very small amounts of starting DNA. You don't need much at all. So these particular genetic tests that you can use commercially, you just essentially scrape the inside cells of your cheek. The amount of DNA that you get from buccal cells from your cheek, that's more than enough DNA. That's how sensitive this test is. So polymerase chain reaction is going to utilize the fact that very small pieces of DNA, these oligonucleotide primers, can bind to your gene, and you can amplify that piece of DNA between the 2 primers. You can amplify it to the degree where you can easily analyze it. So I have another video to show you essentially how polymerase chain reaction works. So let's go ahead and take a look at this particular video. These speakers distort a bit. I turn them down, but then you can't hear it. Let's see if no, that's not that loud. Well, hopefully this will be a little easier to understand. That's a little loud.
The polymerase chain reaction, or PCR, is a technique that can produce over 1 million-fold amplification of target DNA within a few hours. PCR requires specific oligonucleotide primers and plenty of nucleotides for DNA polymerization. It also requires a heat-stable DNA polymerase because PCR requires repeated cycles of DNA solution to 95 degrees Celsius. The polymerase test from the thermophile ferment aquaticus is often used for this purpose. In PCR, the reaction mixture is heated to 95 degrees Celsius for 30 seconds to denature or separate the DNA strand. The temperature is then lowered to 55 degrees Celsius for 30 seconds, which allows the oligonucleotide primers to anneal to known DNA sequences flanking the target DNA. The oligonucleotide primers, which are usually 20 to 30 nucleotides in length, are made so that they're complementary to the flanking sequences. The temperature is then brought to 72 degrees Celsius, which is the optimal reaction temperature for Taq DNA polymerase. The polymerase uses the primers as starting points for DNA synthesis, adding one nucleotide at a time to create a complementary strand of DNA. The cycle is repeated, starting with the 95 degrees Celsius heating step. Cooling the solution to 55 degrees Celsius allows the primers to anneal. Heating the solution back to 72 degrees Celsius allows polymerization to proceed. After 2 cycles, 4 double-stranded target DNA sequences exist. After the 3rd cycle, 8 copies exist. After the 4th cycle, 16 copies exist. After 25 cycles, about 30 million copies of the target DNA exist. Because PCR reactions require 25 to 30 heating and cooling cycles, a machine called a thermocycler is used to reproducibly and rapidly deliver these cycles.
So that's PCR. PCR is the mainstay. Frankly, it's the industry standard for analyzing DNA. The downside to PCR is that you have to know the sequence of the DNA you're going to analyze. Now with the Human Genome Project, we know the sequence. So because of that, you can design primers. Many companies have made kits with specific primers in the mix, and you can use these kits to analyze human DNA for mutations in gene X, gene Y, gene A, B, C, D, E, F, G, blah, 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 whatever, and so on and so forth. Once you amplify the DNA, you can sequence it. In fact, there are kits that will let you go straight from PCR amplification to sequencing. There are even reactions now that do PCR amplification and sequencing at the same time, now with high throughput sequencing. And this takes the time required essentially for this genetic test and makes it very, very short. Because it makes it very short, there's less work, therefore the test is cheaper. Therefore, many of these tests really come down in price, and they're much more doable, much more convenient. You get an answer much more quickly, usually, with these tests. And you don't have to take blood for these, just cells from the inside of the mouth. You take cells in the inside of the mouth, put it in a test tube, send it to the lab with the instructions. It's really that easy. So many of you will probably be asked to get some samples for a genetic test from patients. And you'll just go in with a little swab and you'll say, open up and hit, and then you're done. And that's essentially how the test is largely done. And that they'll be subjected to polymerase chain reaction. Now, the inquisitive parent or child may ask you, what's going to happen to my cells? So it behooves you to be able to understand something about polymerase chain reaction so you can explain it to them in plain and easy to understand language. So you can assure them of what's going to happen to it. They're not going to use it to clone an army or something like that. No, we're just analyzing the DNA. So you can use these types of tests for detection of diseases before someone experiences symptoms. Might that be useful? Sure. Polycystic kidney disease, one of the first signs is blood pressure that just really starts to rise very gradually. If you put them on blood pressure medicine particularly certain types of blood pressure medicines, you can spare kidney function for a while. Will decrease their need for a kidney transplant. Huntington's disease tends to have a very gradual onset as well. There's some medications that can, it seems, delay it just a bit. Also, the family will know what to expect as this person starts to lose certain types of neural functions. So it's not just for the patient, it's also for the support staff as well, family and so on and so forth. You can also screen prenatally for things like cystic fibrosis or various types of thalassemias, alpha or beta thalassemias. These are diseases of hemoglobin. So the patient does not make normal hemoglobin, or they don't make enough normal hemoglobin. And very often bone marrow transplants might be necessary to help this person make enough red blood cells to be able to live. If you have that kind of time, you can identify a donor and so on and so forth. You can screen for X-linked conditions, hemophilia, Duchenne muscular dystrophy, there are lots of others. You can also screen for things like familial polyposis coli, where you have a very, very high risk of colorectal carcinoma. In cases like this, a preemptive removal of the colon essentially can spare someone essentially life-threatening colorectal carcinoma. Some tests are not genetic tests, but they're biochemical tests. So with phenylketonuria, that's a member of this group of genetic diseases called hyperphenylalaniemias. Mutations experienced by individuals who have hyperphenylalaniemias tend to be single base mutations. Those are harder to detect using something like PCR. But fortunately, because people with phenylketonuria have a defect in phenylalanine hydroxylase, they'll have an accumulation of phenylalanine. That's fairly easy to detect. If you detect it early, you can put the infant on a fairly restrictive diet that restricts the intake of phenylalanine. That will prevent the severe mental retardation that results from excessive phenylalanine that interferes with neurotransmitter production. Cystic fibrosis is fairly easily detected using something called the sweat test. The sweat test. And if you work in pediatrics for any length of time, you'll see the sweat test. Individuals with cystic fibrosis sweat enormous amounts of chloride. So there are specially prepared papers where you simply have the patient, usually a child, they put their hand on this piece of paper and they keep it there for a minute or two. When they lift their hand off, if they have cystic fibrosis, there'll be a dark outline where their hand was. If they don't have cystic fibrosis, you won't see the dark outline. That is a much easier and much simpler test than a genetic test. Now, having said that, there are very good genetic tests for cystic fibrosis. Because the CFTR gene, it's a big gene, the sequence of it is very well known, and the alleles of cystic fibrosis are extremely well understood, and there are very good tests for it. But the sweat test is a lot cheaper, and it's a lot faster. Granted, if someone is positive for a sweat test, you'll usually order a genetic test to confirm it. But the sweat test usually will tell you this person is excreting way too much chloride. That's usually due to a defect in the CFTR gene, which is a regulated chloride channel. Again, you know what to expect. You're going to have thick mucus that's going to obstruct the airways. It's going to obstruct the pancreatic ducts and things like that. And that's an example of one test that can be detected with biochemical means. All right, so these types of genetic diseases are so-called inborn errors of metabolism. So the state of Georgia, for example, will screen newborns for 24 different metabolic disorders plus sickle cell disease. I don't know why your book calls it sickle cell anemia. It's not called that anymore. It hasn't been called that now for a few years. It's called sickle cell disease, not sickle cell anemia. State of Georgia is a bit on the heavy side when it comes to required genetic testing. Not all states will test for the same disorders. Now, patients can refuse to have the test done. Typically, what will happen is that some blood will be drawn, and if, say, you're the nurse and you draw the blood, you say, your child is going to be tested for these genetic diseases. Do we have your permission? The answer is yes. Okay, then please sign here to give us, showing that you've given permission, essentially. And then you'll have to sign it as the witness. Some states require 2 witnesses. Michigan just requires the patient and the witness, usually the nurse or the doctor, or something like that. Oh, what state is it requires 2? I think it's Connecticut requires another witness. So usually you sign it, and then they send you out to the desk to have the person at the desk sign it as well, or the doctor signs it, or something like that. So these types of tests, you have to have parental approval in order to run the test. Now, they may say, sure, they can do these 23 tests, but not this 24th test or whatever. There's usually a box at the bottom says test all things. And then the boxes that they don't want, you tick the boxes that they don't want. Or there's another box that says no tests have been authorized. You tick that, and then, sorry, British word, check the box. And then, you guys remember that term, tick the box? No, that's British for check the box. That's what the British say. You tick the box instead of check the box. So anyway, you check the box at the bottom that says test, no tests authorized, and so on and so forth.
So why do they require them to be tested?
So in the state of Georgia, a large population of the state of Georgia are descended from slaves. And the slave population, the part of Africa that they were taken from, there was a relatively high frequency of things like sickle cell disease, glucose 6-phosphate dehydrogenase deficiency. These are all things to protect against malaria. And so because of that, these particular genetic diseases are at a high enough level to where if you detect them early, you can take some preemptive and preventative steps in the life of that child to prevent them from having the sorts of anemic episodes that these sorts of diseases can cause. So for example, with glucose 6-phosphate dehydrogenase deficiency, certain types of drugs or events, the red blood cells are fragile. They can all of a sudden undergo this big burst of hemolytic anemia, and that's going to clog up your spleen, and it's going to clog up your kidneys, and we can assert that's very bad. So you want to try and avoid it. You want to try and avoid sulfa drugs, certain types of foods, fava beans, things like that can cause those types of things. That's probably why the state of Georgia said, oh, let's just go ahead and test that, because it's going to save the hospital's money. Because later on, oh, this kid's got this wild episode of hemolytic anemia. Well, we didn't know. Okay, now you know, you can be preventative about it. The parents are happier, the kid's happier, and it saves the hospital money as well. So you are entering a field that's a business. Like it or not, it's a business. Your job is to render the best care to that person, regardless of whether they're a criminal or a person. I mean, you don't have to like them to give them the best care possible. You might really hate their insides, frankly. They might be mean to you and nasty and snap at you and call you names and say you stink and stuff like that, but you still have to give them the best care you can. That's your job. And that's your calling as a nurse, as a healer, right? And there'll be things about the job. You'll come home, you'll complain to Mike and say, sometimes this job sucks. there'll be other days you'll be on top of the world. So it's just the way it goes. And there'll be things the hospital will get notes, they have to curtail costs because taking care of people costs money and cash flow is an issue. And you'll just say, okay, this sucks, but it's a fact. It's just a fact. And don't think that socialized medicine companies, nations of socialized medicine don't have the same problem. They do. They just solve it in different ways. They ration care. In Great Britain, if you're older and you get cancer, bend over, grab your ankles, kiss your butt goodbye. You're not getting care. That's the way they save money. And don't let anybody tell you differently. Oh, they don't ration care over there. Yes, they do. Yes, they do. Canada does the same thing. That's how they save money. Very good. So here's our summary of chapter 2. A family history is worth its weight in gold. Worth its weight in gold. And in fundamentals, you'll learn a lot about how to do a good medical history, pay attention, and make sure that you become very good at taking a medical history. First degree relatives, parents, siblings, offspring, so on and so forth. That's going to be kind of your money group when it comes to genetic disorders. If possible, more distant relatives if they know about them. And so that pedigree can be used essentially to analyze mammalian inheritance of certain traits. Now, case study number 1 is a example of a husband and wife, an interview essentially with a husband and wife. You're taking a pedigree and you get answers and you discover their genetic diseases in their family's history, and they want to know about the risk to their baby that they're going to have. And so after the exam on Monday, I'd like you to work on case study 1 and get an idea, take the information there and make a pedigree and figure out the risk essentially that the couple wants to know that their baby's going to have a certain type of genetic disease. So that will be an assignment you'll have to do after the exam, but focus on the exam for Monday, and then you can work on case study 1. Cytogenetic tests can be ordered and they're used to study chromosome structure and chromosome numbers. So gross chromosomal mutations can be in structure or numbers. Cytogenetics can reveal these if they're big enough to see at least. For particular genes, you're going to use things like PCR or things like that. Other tests, however, are best detected with biochemical testing. These types of genetic conditions are in the minority, but where there are good biochemical tests, those are usually used because they're much cheaper, much cheaper. Very good. Any questions about chapter 2 before we move on to chapter 3? No? All right, let's go ahead and have a look at chapter 3. All right, now with prenatal genetics, unless you're doing amniocentesis or chorionic villus sampling, you're usually dealing with a couple who are using in vitro fertilization. They're a couple, they're either older or they may have some sort of history of genetic diseases and they want to try to have a child who does not have a particular genetic disease, or maybe they're using donor eggs or something like that if the woman's ovaries don't produce eggs, or maybe they're using sperm from a sperm bank or something like that. So this essentially is what this chapter is about. So some of you may be aware, I published a book several years ago called Stem Cell Epistles. I have a chapter in there about in vitro fertilization, and that goes over a lot of this if you want to read it. I can get you a copy. I'll even sign it. And that you can read that chapter if you want. It's available on Kindle as well. So when you refer to a test, it's not necessarily a diagnostic test. You're essentially screening embryos that have not yet been implanted into the mother's uterus yet. Now, screening can also include ultrasound examination of babies. You can also have maternal serum screening. And then there are some examples of non-invasive prenatal screening that we'll talk about as well, others of which that are a bit more invasive. Ultrasound, of course, is pretty non-invasive. Have any of you, do any of you have siblings you've gone to the ultrasound of your little brothers or sisters or anything like that? I still remember the ultrasound of our first baby. Ultrasounds now, my word, the technology has run. It hasn't walked ahead, it's jumped ahead. The clarity you get with an 3D ultrasound now is remarkable, absolutely remarkable. And the ultrasound machines too have gone from being these ponderous things that were as big as this table to things now you can use at the bedside. So ultrasound is used a good deal. No evidence whatsoever that ultrasound is deleterious to the baby at any stage. Then maternal serum screening, and then various types of non-invasive screening. I'll look at those. So ultrasound is used to examine the structure of the baby at different stages during development. Very often, if a woman thinks she's pregnant, and she usually thinks she's pregnant when she misses her period, by that time, the baby is already 2 to 4 weeks old. And to just give you a highlight, essentially, by 4 weeks old, essentially most of the organ systems of the baby are either mostly formed or on their way to being mostly formed. They're vastly immature, but they're mostly formed. So let me first designate you between these two terms, fetus and embryo. So when we talk about an embryo, so embryo is usually reserved for the first 8 weeks. That's the first 8 weeks after fertilization. When you look at OB books, they'll usually time the fetus from the period of last menstruation. So it'll be usually, this will be 2 weeks behind that. So time of last menstruation, in that case, would be the first 10 weeks. So if you're doing this from the time of fertilization, 8 weeks, from the time of last menstruation to be 10 weeks. So essentially 56 days. After that, the baby is technically a fetus. Technically a fetus. And by the time the mother has an inkling that she's pregnant, usually the baby is already well within halfway through the embryonic period. So with an ultrasound, there are several things you're looking at. One of the first things you're looking at is the thickness of the skull. Down syndrome babies, you look at a thing called nuchal thickness. So the nuchal area is back here. So you have this little nuchal protuberance here. That's for attachment of the nuchal ligament, where the trapezius and other muscles attach, and that helps, you know, raise the head and so on and so forth. That is very thick in a child with Down syndrome. It's thicker than normal. So the nuchal thickness is quite important. And that is essentially nuchal translucency, essentially. So if you have a thickening of that nuchal translucency, that's usually associated with Down syndrome. Now, the other thing you're going to look at is head to rump, crown to rump, excuse me, crown to rump distance, and then also the diameter of the head. That's going to help you tell the age. And then from that age, you can predict the due date, of course. So if a mom comes in and the baby is 9 weeks old, based on development, pregnancy is about 40 weeks-ish. So she has another 31 weeks to go. So you count 31 weeks from 9 weeks and you get a due date, essentially. Due dates are always are good estimates, aren't they? Trust me, they're estimates. They're estimates. Some babies are small, but remember, it's late in the third trimester when you get most of the growth. So usually at this early stage, all babies are about the same size. The difference in growth usually starts to come within the third trimester. And so some women, for example, come from families that have small babies. And so, case in point, I come from a family of small babies. My wife comes from a family of small babies. So our oldest was 5.5 pounds at birth. She was completely full term. She had long hair. She had long fingernails. She was full term. She was estimated as being problematic because she was small. And then the OB finally said, "This baby is just small." To which my wife said, "Yes." She didn't want to give birth to a really big baby. These kids with, 11-pound babies who give birth to toddlers. No thanks. So, yeah, 11 pounds. Yeah. My uncle was an 11.5-pound baby. Yeah. Ugh. She can. She can. Yeah. Yeah. I'm going to give birth to a bowling ball. Yep. So now, because when you do false color with ultrasound, you can use false color. That false color will help you detect organs really easily, and you can detect abnormalities, structural abnormalities in certain organs. A good ultrasound tech can just see them right away, essentially. Sometimes things that you see on ultrasound end up working themselves out in development, other times not. So just because a couple gets some potential bad news on an ultrasound, it's not ineluctable that they will then have a baby who has some sort of structural abnormality. So something to keep in mind. Now, genetic screens essentially can assess for potential genetic diseases that may cause certain birth defects. And there are certain structural features of a embryo or fetus that can tell you essentially if you have some sort of genetic issue, usually. And there's a nice proviso there at the bottom. Normal ultrasound does not guarantee the birth of a healthy baby. There's just stuff that can go wrong during development. There's stuff that can go wrong during birth. Keep in mind, I don't know if you learned this, you probably learned it and then probably forgot it after the test, but during pregnancy, a woman, the number of her clotting factor levels go up quite a bit. So her clotting time goes down. That means she's at increased risk for clots pregnancy is a hypercoagulable state. So she can get clots in the umbilical cord. That can cause ischemia. The baby can die. The baby can have cerebral palsy because of that, so on and so forth. So these are all things that can potentially go wrong. So there's no guarantees. There's no guarantees. And unfortunately, healthcare workers in hospitals get sued because something goes wrong, and people have this notion that they have a right to a completely healthy baby. Do they? No. You have a right to anything in this life? No, we don't. And when we demand certain rights that we actually don't have, I think that's when things start to really fall off the track. Okay, other types of tests that you can analyze, these are so-called analytes. These are things that you find in the amniotic fluid. And they can be indicative of certain developmental abnormalities. In particular, open neural tube defects. So open neural tube defects, just quick review of embryology. The neural tube folds up, and then you have neuropores at both ends, front end, back end. The front neuropore closes about day 22 of development. The back one closes about a day later, about day 23, 24 of development. If the back one doesn't close, you're at increased risk of spina bifida. If the front one doesn't close, you're at increased risk of anencephaly. Anencephaly. So anencephaly, essentially, this part of the head is missing in the baby. You just basically have a brain stem. The heart is still beating because the brain stem's there, but there's no brain waves, there's no EEGs, and those children don't last long after birth at all. The redeeming factor of having an anencephalic kid is that they're very good organ donors for kids who need organs, because the organs are small and they're usually quite healthy, and they can be used for that. So those are the two main types of open neural tube defects. Other defects that you can see is if the vertebral column doesn't close all the way. In fact, those of you who do labor and delivery, one of the tricks you look at when you look at the back of the baby, if you see a tuft of hair on the back of the baby, that tells you the vertebral column's not closed, because the neural tube, which is an extremely powerful signaling organ, as well as being the early nerve cord, that will actually induce the growth of hair on the back. The vertebral column normally says, okay, signaling molecules, you just stay here. We're not going to have hair on the back. But if you see a tuft of hair, that's an indication you have an incomplete neural tube. Now, if it's really open, then you may see essentially the tube, you may see like a little blister thing on the back where the tube is actually out of the vertebral column, and that has to be surgically repaired. And the nerves may be so abnormal that child may never walk, but usually you can repair that surgically. All right, so some of these markers, as you see, can be indicative of certain genetic conditions. So Down syndrome, as you can see, can be indicated by increased levels of human chorionic gonadotropin. That's a hormone that's made by the placenta. In fact, it's made by the embryo before it ever makes the placenta. It signals to the mother before attachment ever occurs. The embryo and the mom are constantly talking to each other. There's this line of communication that goes on between the two. That's why I find people who call babies parasites on their mothers to be remarkably uninformed. Parasites take, they don't talk. The baby is not a parasite, it's a welcome guest inside the mother's body. It's exactly where it was designed to be. It's not a parasite. Yes, it draws resources from the mother. So do toddlers. So do toddlers. You disagree with me? Try being around one all day and you'll see. This particular protein here is so-called pregnancy-associated plasma protein. Its levels decrease, usually slightly, but in some cases pretty substantially in Down syndrome. But in cases of trisomy 18 or 13, it's usually down quite a bit. So that ends up being a pretty good marker for these trisomies. Alpha fetoprotein is an extremely good marker for certain types of genetic conditions. Now, it says when it comes to the open neural tube defect that it's not very informative. I would dispute that because alpha fetoprotein levels are usually quite high in individuals with anencephaly. Alpha fetoprotein is also a very good marker if you have a baby whose kidneys didn't develop, so-called renal agenesis. And you can see that these levels may change a bit in the second trimester. So Down syndrome, you can see the pregnancy-associated protein will go down then. These others here, inhibin, UE3, alpha fetoprotein, so on and so forth, they will tend to change sometimes in some cases. Here we go. Later on, you can see here, increased levels of the alpha fetoprotein are a very good marker for open neural tube defects. Sampling the amniotic fluid essentially can detect these. You can also detect them to some degree just from a blood test. So these can be detected very nicely with blood tests. And so first trimester screens can detect essentially these proteins, and they give you a profile of per risk for these two, trisomy 18, 13, and Down syndrome, trisomy 21. And you can take these other factors into consideration. These particular genetic conditions are usually found at a higher frequency in mothers who are insulin dependent diabetics. Albeit, if the mother controls her blood glucose levels well, those risks can be mitigated to some extent. The big issue with insulin dependent, I wish they wouldn't say insulin dependent, type 1 diabetes, is if you have high blood sugars late in third trimester, the baby will usually tend to get really big, and you may give birth to a very large baby. 12 to 13 pounds is not unheard of, which at that stage is just give up and give me a C-section. So by combining the biochemical screens with the ultrasound screens, this turns out to be a very powerful way to screen for these early genetic conditions. Very powerful way to do that. Now, trisomy 18, trisomy 13, those children usually, they may be born alive, but they will not survive long, typically. Down syndrome children, of course, have a huge range of capabilities, disabilities, so on and so forth. And they may be very severe and they may not live very long, or they may die before they're born, or they may survive and flourish. So if you work peds, you'll see a wide range of Down syndrome children. There was a guy, when I was at UC Davis, there was a guy who had Down syndrome. He was a computer science major. He's a computer programmer today. Yes, he has Down syndrome. He's obviously not very severe. Then you have kids, if you go to Lyle Torrent in Jackson, there are kids there who have Down syndrome who are very severely affected. So it's a range. It's a huge, huge variation in Down syndrome kids. So they vary, and they vary remarkably. So this one screen for second trimester markers is called the quad screen. Some hospitals will do the quad screen if you're an older mother, which nowadays is most people. People just waiting longer to have kids these days. So quad screens are very, very common. Yeah, Manny? 30, above 30 usually. Once you hit 34, past 34, your risk of Down syndrome now starts to climb substantially year by year. Having said that, so Andrew Fitzpatrick, he was on the tennis team here. He married his college sweetheart, and his mother, Joan Fitzpatrick, was the elementary school music teacher in the district here. And Joan adopted a Down syndrome child named Michael. And so Andrew essentially grew up taking care of Michael. And Michael and Andrew were buds, complete total buds. He then and his wife had their first baby, and they had a Down syndrome child. And she was young. She was maybe 2-3 years older than you, and they had a Down syndrome child. So Patrick said, "No, I got plenty of practice looking at working with Michael." And their child, unfortunately, is quite severely affected. He's got heart trouble. He's got the beginnings of leukemia, but he's a fighter, and so are his parents. So sequential or integrated screens, these are usually batteries of screens that you can order or pre-order for a family. And that will essentially include the first and second trimester set of screens. Again, you're screening for Down syndrome, trisomy 18, trisomy 13. Why are you screening for these? Because those are the most common genetic conditions. And then, of course, the open neural tube defects, either spina bifida or anencephaly. This type of screening has a very high detection rate. So you have 2 blood draws, one in the first semester, one in the second semester, and then the usual ultrasounds, albeit you will usually have ultrasounds earlier than you would normally have. Instead of having them anywhere from 10 to 12 weeks, you might have one a little earlier. Now, you may have heard, if you haven't, then I'll just tell you, the baby and the mother during pregnancy exchange cells. So for example, there's a very famous case of a mother who had a heart attack while she was pregnant. She was treated, let go from the hospital. She recovered, she gave birth, and then during her autopsy, it was discovered that there was a patch of tissue that had healed the heart attack tissue, and that tissue genetically had come from her baby. And since then, there's been a wide range of screenings. Turns out mothers have bone marrow cells that came from their baby. They have cells in their kidneys that came from their baby. They even have brain cells that came from their baby. So it turns out that because there's an exchange of cells, mothers have DNA in their blood from their baby. And these types of non-invasive prenatal screening tests, cell-free DNA, you're looking for the baby's DNA in the mother's blood. You don't have to have some breach of the placenta. It just happens normally. Again, the mom and the child, they're talking to each other. They exchange cells with each other. And some of the mother cells are also found in the baby. So again, this business that the baby is, no, I'm not going to get off this hobby horse. These people who say babies are parasites on their mothers are idiots. They don't know what in the world they're talking about. Baby is not a parasite, it's a welcome guest. So these types of tests, they can give you a risk profile for trisomy 18, 13, and Down syndrome. Also, you can look for aneuploidy of the sex chromosomes doing these tests. You get a very high detection rate, low false positive, than you do with the maternal serum test. These tests have also come down in price substantially since they were first introduced. It doesn't replace the other tests, but some physicians will recommend these tests if there are questions about the results of the earlier tests that we talked about. So this is a table that comes from chapter 3 of your book, the McClory book. And so this just shows you the detection rates, false positive rates, so on and so forth. So if you're using biochemical tests only, your detection rate is 60 to 70%. So you're missing 30, 40% of the cases. That's not great. That's kind of sort of okay, not great. And then the detection rate of trisomy 18 or 13 is about the same. So overall, for using just biochemical tests, yeah, just okay. You don't really get a good detection of your open neural tube defects. False positive rate is 5%, pretty good actually. Again, not great, but just good. Now, if you combine this with ultrasound, now look essentially at your detection rate. Now you're getting 91 to 95% of detection. Much better, much better. Still not able really, at least in first trimester, to detect the open neural tube defects. False positive rate is still about the same. Quad screen, using a quad screen, about 80%. Now you can start to detect the open neural tube defects. And if you use a sequential screen, now we're starting to really rock and roll. This is quite powerful. And then the integrated screen really is your best bet. Some insurance policies, you can actually buy policies that will cover the integrated screen. And typically they're what insurance calls add-ons. So for example, our insurance here, say for example, let's say McKenna graduates, she comes back and she teaches for us. Let's say she and Michael want to have 3 kids? 3, okay, 3 kids, you should have 4. You thought about it, think harder. Say they want to have 3 kids, let's say. So they think, let's say, oh, there's something on your uncle's side or something that's kind of questionable. So they can actually get an add-on to their policy, and it'll bump their cost up every month a bit. But if all of a sudden she gives Michael the good news, now they can get this screening and they don't have to pay extra for it because the policy paid for it. And then once they're done having their family, then they can say, okay, let's not do that. We don't want to pay for that because we've got our 3 or 4 kids, and we're done with this. And that sort of add-on gives you the possibility essentially of having this and then not have to, oh, gee, do we want to shell out that much money for this or something like that. So some hospitals too will recommend these sorts of things depending upon the risk profile of the mother or something like that. Ooh, okay. Why don't I stop right there? Okay, well, after Monday, we'll talk about the chorionic villus sampling, amniocentesis. Then we'll get into pre-implant genetics, and that gets into some definite hairy, ethical, sticky wickets that, well, maybe might want to talk about. So 50. So guys, we're short on questions. So get a writing because I'm going to write the test this weekend. You do?
Writing some extra one.
Okay. Make sure I have plenty to choose from, okay? Okay, folks, you two have a great weekend. And Hannah, you're still on crutches. Are you able to come in for the exam, or would you rather make other arrangements?
I think on Monday I should be good to take the exam, but I was gonna ask you, do you have a class between like 11 and 12?
I do not.
Okay, is there any way I could take the exam early just because I have an appointment with my surgeon at 2:40? So I'm gonna have to get to the hospital at 2. they
Yes,
said.
there is. Would you like to take it at the academic connection? Student
Yeah,
connection?
that's totally fine. That's totally fine.
I'll send Amber a copy of the test once I get it written. You will then need to make an appointment
Okay.
with her. And I have to tell you, her appointments are filling up. So make it soon.
Okay, thank you.
All right. You have a great weekend, and hey, nurse that knee.
Thank you so much.
Bye.